Okay, good morning everyone. So today we're going to do a bit of a recap on what we know so far on photosynthesis, and then we're going to focus on the Hill reaction. Okay, so we know so far, so green parts of plants, this is where photosynthesis takes place because of iodine experiments. We narrowed it down to chloroplasts. We know that this is the structure of a chloroplast. We know that in the chloroplast we have thylakoids, and in these thylakoids we've got photosystems, and this is what a photosystem looks like. We said that chlorophyll A is going to accept electrons from a donor and give them to an acceptor. So that's the first idea. Electrons are going to move around. Okay, so photosynthesis occurs in the thylakoids and electrons move around, they are involved. The second thing we know from Rubin and Kammen's experiments is that because of how water was labelled and how carbon dioxide was labelled, they were able to do this hypothesis that photosynthesis is two things. The first one being the oxidation of water. So water is transformed into O2 and 4 H pluses, 4 electrons, and on the other side it's coupled with the reduction of carbon dioxide. So CO2 is reduced into CH2O plus H2O. So what's interesting is that we've said that in the photosystems electrons are involved. We know that in photosynthesis, thanks to Rubin and Kammen's experiments, we can say that we probably have these oxidation and reduction equations. So again, we have electrons here and there. So basically, the experiment we're going to have a look at today is a proof that these oxidation and reduction reactions do occur. It's thanks to this experiment, so Hill reaction, that we know that reduction and oxidation occur. Okay, so here we go. That was for the recaps. Okay, so 6.2 Hill's reaction. So this name Hill comes from a chap named Robert Hill. So here's a picture of him. So he was quite an important scientist in the 20th century, and he worked in one of the biochemistry labs in Cambridge, and he did a lot of work on photosynthesis, and especially on all the part which deals with these electrons. So you had back exercise to look at. We're going to study this back exercise, which is based on Robert Hill's experiment. So here we go. Okay, so Robert Hill published in 1937 results from a series of experiments on photosynthesis. Okay, isolated chloroplasts are placed into water without CO2. So that's our two first elements. We've got isolated chloroplasts in water without CO2. Okay, so let's have this. So basically we're going to have a broth with isolated chloroplasts. So let's add a few chloroplasts into this. You may want to add onions and carrots for taste, but that's not going to help our experiment at all, so we're going to keep just chloroplasts. Okay, so what's important is that there is no CO2. So we can cancel out CO2. We know that in this experiment there is only water, dioxygen and chloroplasts. Okay, that's the first thing. And the second thing, so that's new for you, there's this molecule called DCPIP. So it says it's an electron acceptor, that means it can take electrons, it can gain electrons. And this is interesting. The chemical is blue when oxidized. So oxidized means it doesn't have these electrons and H pluses, so it's DCPIP, and it's colorless when it's reduced into DCPIPH2. Okay, so this might be a little bit obscure, so I'm going to try and detail it a little bit more. So first thing, why is it called DCPIP? That's just the main letters of its name. You have them here. So D, C, P, I, P. There we go. So the full name is 2,6-dichlorophenolindophenols. You don't need to know the name. You just need to know how this electron acceptor works in this experiment. Okay, so this molecule looks like this. Okay, so this is the oxidized version of the molecule, and let's write it here. So oxidized, and when this molecule is oxidized, it's blue. Okay, so here we are. So this oxidized version, DCPIP, it can be reduced. So when you have a reduction, if you remember, you have a gain of electrons. Let's add, say, 2 electrons two protons. Oops, forgot the 2. Okay, and this is going to give us, so this molecule plus 2 electrons plus 2 protons will give this molecule, and you can see that we've got 2 extra hydrogens, one here and one there. So the molecule is still neutral because we've also added 2 electrons. And this one here, this molecule here is colorless. So just to recap, this molecule, so DCPIP, is blue. It's the oxidized version. When you reduce it, so you add 2 electrons plus 2 protons, you get this molecule, and this molecule is colorless. Okay? So let's move on to the more detail of the experiment. Okay, so we've got 3 tubes. In each tube we've got, so what we said earlier on, water with without CO2 and the isolated chloroplasts. Okay, so that's the first thing, and then it mentions if there is DCPIP or not. So tube 1 there isn't, and there are DCPIP in tubes 2 and 3. So let's make the diagrams of these tubes. Okay, so tube 1, as we said, chloroplasts and water, no DCPIP. As chloroplasts are green, the solution is going to look green. So I'm just adding colours for illustration. So as the chloroplasts are green, that's why the tube will look green. Okay. Second tube, we've got the chloroplasts, but this time we've got the DCPIP molecule. This molecule is blue, and it's going to cover the chloroplasts which are green. It's going to have a more powerful dyeing power, so that means that the tube will look blue. That's why the tube is blue. Same thing for tube 3, the DCPIP will have a more powerful dyeing colour. That's why the tube looks blue. Okay, and as we said, in all of the tubes there's no CO2. That's for the explanation for the colours. So tube 1, just the chloroplasts that's why it's green. Tube 2, chloroplasts plus DCPIP, that's why it's blue. And tube 3 is the same as tube 2. Okay, so we've done this part here. We've explained what we have in the tubes. Okay, second part of the experiment. They're going to be exposed to light. Tube 3 is going to be covered with aluminium foil, so the tube will be covered and won't let light go through. So that means that tube 1 and tube 2 are going to be exposed to light. So I'm doing this part now. So tube 1 and tube 2 are going to be exposed to light. So same thing, tube 1 and tube 2, they're going to be exposed to light, whereas tube 3 is going to be covered with aluminium foil, so no light can penetrate. Okay, and let's look at the results now. So tube 1, it's still green, so no change compared to the beginning of the experiment. Okay. Tube 2, this is interesting. Tube 2 went from a blue colour to a green colour. So we're going to have to explain this. Okay. So we can say that in the presence of light, tube 2 went from a blue colour to a green colour, and we'll have to explain that. So in the presence of light, tube 2 went from a blue colour to a green colour. And tube 3, so it wasn't exposed to light and it stayed blue. So we can clearly see that light has an impact. The only difference between tubes 2 and 3 is the presence of light, and we can see that in the presence of light there has been a change in colour from blue to green. So we're going to move on to the next slide to explain why there is this change in colour. So as a reminder, that's what we have in the tubes. So tube 2, at the beginning of the experiment, we have it here, we've got chloroplasts and we've got DCPIP, and you remember that DCPIP is blue, that's why the tube has this blue colour. We know that with light, so when it's exposed to light, there is a change. We go from a blue colour to a green colour. So this means that DCPIP has lost its blue colour. And we know that's possible when it's been reduced. When DCPIP has been reduced to DCPIPH2, it has no longer its colour blue. So that's exactly what's happened. So now we can be a bit more precise. We said earlier on that because of light, tube 2 was blue and became green. Now we can be a little bit more scientific. In the presence of light, DCPIP has been reduced to DCPIH2. That's what we just saw earlier on. So in the presence of light, I'm just going to put a nail for light here, DCPIP, which is here, has been reduced to this molecule here, DCPIH2. So I'm just going to write them. So DCPIP, as I said, in the presence of light, has been reduced to DCPIH2. Okay, and we could maybe even go further. We could maybe make this hypothesis that light intervenes in mechanisms which releases electrons. Otherwise, DCPIP would not be reduced, because to be reduced you need the presence of these electrons. So just to recap the 3 steps, first thing which is obvious is that we go from a blue colour to a green colour in the presence of light. The second thing we can say is that, which is more scientific, is that DCPIP in the presence of light has been reduced to DCPIH2. And the third thing we can say as a hypothesis is that because of light there is a mechanism which releases electrons, and these electrons are then taken by DCPIP, which is an electron acceptor, and therefore the molecules are reduced into DCPIPH2. So what we learn from this is that light provokes the release of electrons and the reduction of some molecules. In this case it's DCPIP. We'll see that in plants there is an equivalent to this DCPIP. DCPIP is a molecule that Robert Hill added in the experiment, and we'll see that there is a similar molecule in plants. Okay, so that was for the general explanations about the experiment. For it to be a little bit clearer in your notes, I've just taken one by one the back questions, and I've copied some of the answers from the official marking scheme. So we're going to look at that so that you have first the general explanation and now detailed answers if you were to answer these questions again. Okay, so we're now going to tackle these 4 questions here. 1, 2, 3, 4. Okay, first one. Well, basically we've done this. We've explained tube by tube. So I think the easiest is just for you to read this paragraph. So what you can do is obviously pause the video and write the answers. But normally you should find out what you see in these answers is basically what we've been saying throughout the explanation of the experiment. Okay, maybe there is one thing that we didn't say is that so the mechanism I said earlier on is going to be part of something which is called the light phase of photosynthesis. So I'm not going to give too much for the moment, but what you know is that electrons are produced during this light phase of photosynthesis. So as I said, we're trying to build this gradually. So we're starting to understand that there are 2 stages, and one of the first stages is called the light phase photosynthesis, and electrons are produced. But we will have general diagrams to recap this, so we're not going to go too much into detail on this light phase of photosynthesis. Okay, so I'll let you write and go through these answers. Obviously, if you have questions, you can always email me or pronote me on these answers. Okay, question 2. Explain why in this experiment the O2 released does not come from CO2. This question is not directly aimed on this experiment really. It actually links with what we've just seen before. So we said that in this experiment there's no CO2, so if there is dioxygen that is produced, it can't come from CO2. In this case it has to come from water, and if you switch back to Rubin and Kammen's experiment on the previous video, you will notice that we said that the O2 comes from water. So this is coherent with Rubin and Kammen's experiment. So basically it can't come from CO2 because there's simply no CO2 in the test tube. So this is the answer. No CO2, CO2 free, so therefore the O2 can't come from the carbon dioxide. And we know that it comes from water because of the Kammen and Rubin experiments. So same thing, I'll let you pause the video and write down the sentence. Okay, third one. So as I said, Robert Hill used DCPIP as a electron acceptor. In actual plants, it's another molecule, and the name of this molecule is NADP. Okay, and we'll see this later on, but if it's a electron acceptor, this means that NADP can gain electrons. So it's going to be similar to DCPIP, plus 2 electrons and 2 protons, 2 electrons and 2 protons, and you're going to end up with NADP2. Okay, so in plants there is an electron acceptor, it's called NADP, and you've got a similar pattern, NADP plus 2 electrons plus 2 protons will give NADPH2. Okay, so same thing, I'll let you write down the answer, and we're going to move on to the last one. So question 4. The initial experiment was repeated, but the chloroplasts were replaced with chlorophyll. After exposure to light, the content of tube 2 is blue. Okay, so instead of having chloroplasts, in tube 2 we simply have chlorophyll. So okay, I'm just going to illustrate with a few lines, but that means instead of having a whole chloroplast, we only have a few pigments, which makes a difference because it is said that at the end, so we've been through the light exposure and the tube remains blue. So what does this mean is that for this light stage to occur, chloroplasts need to be intact, and we'll see this in detail. If you've only got the pigments, it's not going to work. So we'll see that this electron acceptor can only work if can only take the electrons if the chloroplast is intact. So that means that the chloroplast has to be intact for these electrons to be available. So this is the answer. For the Hill reaction to occur, intact chloroplasts are needed. We'll give a little bit more detail to this back question a little bit later, because we need to see a few things inside the thylakoid. So we'll give more information about this question later. Okay, just to recap. So basically, all this experiment has told us two things. Is that in chloroplast there is a mechanism which produces electrons, which releases electrons, and there is something that can catch these electrons. So in this experiment it was DCPIP, in actual plants is NADP. So first thing, something releases electrons, produces electrons, and second thing, the name of this is part of what is called the light stage of photosynthesis. So we're going to stop here for this video. In the next video we're going to try and assemble all these pieces to really explain what is happening during this light stage in the chloroplasts, and then we'll see later on that there is a other stage separate from this light stage. So as I said, we're building this gradually, and at the end with all the diagrams you will understand the whole function of photosynthesis. Okay, have a good day. Goodbye.